Okay, good morning, boys, and we are back to the same chapter. And in the previous sessions, we discussed, I think, all the three experiments related to search for genetic material. In that, first one is Griffith's experiment. It is famous as Griffith's transforming principle. Remember, you should remember it as a rat's experiment or mouse experiment. And this has proved that there is, good morning, this experiment, Griffith's experiment, has proved that there is something that is transforming. But see, he conducted this experiment way back 1928. So at that time he could not use the word DNA. Then to ascertain this experiment, three more scientists conducted one more experiment. That is Oswald Avery, Colin MacLeod and Maclean Maclay. These three conducted what we call biochemical characterization of transforming principle. These three wanted to know what is that Griffith was assuming or thinking of. So they used enzymes here. They used three types of enzymes: proteases to dissolve proteins, and repeated Griffith's experiment. But still, transforming principle was taking place. That means they ruled out proteins as transforming principle. Then they used RNAs, an enzyme that dissolves RNA, and repeated Griffith's experiment. Still, transforming principle taking place. So they said RNA is not the genetic material, transforming principle. But when they use DNAs, DNAs is an enzyme that dissolves DNA. Okay? And when they did this, transforming principle stopped. It did not go. That means they concluded with the help of their experiment that the transforming principle must be DNA. And then experiment number third, which we discussed, Hershey and Chase. This is called blender experiment. Not blunder, blend. No, something that your mummy puts in the mixie to make chutney, like that. So it's a blending. Here the biological organisms that they have taken is bacteriophage and bacteria. You know the difference. Bacteriophage is a virus. Bacteriophage is a virus that attacks bacteria. So here they have used the, again, biochemical component, and they have used radioactive isotopes. In chemistry, I am sure you have learnt what is radioactive substance, right? So two radioactive substances they have used. One is the P32, right? Another is the S35. All of us know in our previous sessions the DNA is made up of three substances. One is the deoxyribulose, which is a sugar, pentose sugar. Second one is the phosphate molecule, P. That is the reason why they have used Hershey and Chase P32, phosphorus 32, heavy dose, radioactive isotope. And third molecule, of course, all of us know, nitrogenous bases. So DNA is made up of, we said, SPN, S sugar, P phosphate, N nitrogenous bases, SPN. Whereas in proteins, we know that portion of protein is sulfur. So they have used S35 as the radioactive substance to identify protein. So when they have done this, using this radioactive isotope, they identified that the outer coat of bacteriophage is illuminating. That means outer coat is made up of protein, whereas the head region having DNA illuminated when it is done with P32, phosphorus 32. That means Hershey and Chase understood that the DNA has got phosphate group in it, and the proteins has got sulfur group in that. They allowed the bacteriophages with radioactive isotopes to infect bacteria, and EG bacteria that they have taken is E. coli, because Escherichia coli is a good bacteria, it's our good friend, it will be present in our stomach and intestine, so easily available. So they made E. coli to be infected with the bacteriophages having radioactive isotopes. After having done that, what they have done? They put it in the mixie blender, okay, centrifugal they have done. When they have done, they identified that the head region having nucleus, having DNA, has got radioactive phosphorus, and the protein coat, outer body of the bacteriophage having sulfur, got radioactive body. So bacteria infected with bacteriophages, that is viruses, having radioactive DNA were radioactive. That means DNA has passed from bacteriophage, that is virus, to bacteria. So this is how Hershey and Chase were credited for having proved beyond doubt that DNA is the genetic material, and that is also what Griffith said, transforming principle, right? So these three experiments are very important, and time and again I said that one of the experiments will be asked as a 3-mark question or a 5-mark question in your board exam. So you must draw the diagram, explain the concept correctly, and if the Hershey and Chase experiment is asked, you have to explain all the 3 steps: infection, second step blending, third step centrifugation, and then finally which the radioactive substance is present in protein, which radioactive substance is present in DNA, whether it is the protein that has come after blending in the centrifugal machine, or is it the DNA. So you have to ascertain that DNA is the genetic material. So once we know that DNA is the genetic material, what are its properties? So genetic material must have the following: ability to generate its replica, means copy. It has to produce its own copy. What happens, Kadkol, if it does not? Sandeep?
It will die soon.
It will die. Who will,
Multiple copies will not be formed.
who will be dying and who will be present?
Virus.
Virus. Now we are talking of DNA. The first property of DNA is ability to generate its replica through replication, DNA replication. Now diagram also you are seeing here. Look at that. I will expand so that you can see correctly. Look at that. Right? So Sandeep, if this does not take place, what will happen?
No gametogenesis.
No gametogenesis. Okay. Next, anybody else? In a cell,
Haploid will not become.
haploid will not become. Okay. See, the clue is Vivek, Kadkol. Clue is a living cell inside that cytoplasm, inside that nucleus, inside that chromosomes, inside that DNA. If DNA fail to produce its own copy, what will happen? That is the question. Yeah, Prasad. Is Prasad available?
Cell division won't take.
Cell division won't take place. Okay. Gollal Gowda. Inheritance.
Cell division will not take place. Same.
Cell division will not take place. Okay. Any other idea? Think out of hat.
DNA will not convert into RNA.
DNA will not convert into RNA. That's why I am showing here. And what is the process called? DNA becoming RNA?
Transcription.
Transcription will not take place. Isn't it? And what is happening? RNA is becoming finally protein. Amino acid chain is protein. That process is called translation. So, if translation fails, no proteins. Sandeep, without proteins, how we will be? How the cell will exist? Understood? So, to form proteins, there has to be replication. DNA must break itself. And remember, when we were talking of anatomy of DNA, I said, between two nitrogenous base pairs, there will be weak hydrogen bond. And I said, little bit energy you supply, hydrogen bond being weak will be broken. And that is enough for us to break DNA. And when we break this double helix model DNA to vertical threads, vertically you break it, that is DNA replication. That is DNA replication. Okay? And once the DNA replication is done, it is formation of RNA. It can form a new DNA also. Isn't it? Vivek? And when cell divides, as Sandeep and Vivek said, if cell divides, the cell, new cells also should have DNA, no? Without DNA, there cannot be nucleus. So cell will die. New cells will not be formed. So for two purposes, DNA replication is a must. One is for the cell to have proteins, there must be DNA replication. For the generation of new cells, there must be DNA replication so that new DNA will be synthesized. Correct now? That is what we said. See, incoming UV radiation, ultraviolet radiation, and look at that, hydrogen bonds between the nitrogen bases broken. Little bit of energy you supply, the nitrogen bases will be broken because of presence of weak hydrogen bond. Right? So that is the meaning of first property. The DNA undergoes replication. And without replication, there will be problem for us, right? And second, chemical and structural stability. Having three chemicals, what we call saturated compounds and unsaturated compounds, you say in chemistry, isn't it? So chemically it is stable, and structurally double helix. So every bond is attached with one atom or the other, hence structural stability also will be there. And then third one, most good question. UV radiation from where? This we are doing it, okay, in the lab. But inside us we have got ATP molecules, isn't it? So those using those energy, ATP molecules, the DNA can be broken. But minimum energy is required. Okay? And next, provide the mutations needed for evolution. See, we said mutation is sudden change that takes place all of a sudden. A change that takes place all of a sudden. That is mutation. Like I said, say a person has gone to bed tonight and tomorrow morning he gets up with a small TT ball on his forehead, with a small extra growth in the arm. This sort of thing we call it mutation. And some mutations are very good mutations, some are lethal, dangerous mutations. And I said, if you recollect from your memory, previous chapter, that one nitrogen base, normally we said one purine should combine with one pyrimidine. Correct? adenine should combine with thymine and vice versa. Thymine can combine with adenine. At I said, or ta. It can be at or ta. Correct? Second combination, guanine with the cytosine and vice versa. GC or CG. If one of the nitrogen bases is not there in this combination, then there won't be any pairing of nitrogenous bases. If the pairing is not done, that leads to new character, new trait, and that is point mutation. If you recollect from your memory, I said point mutation, right? So like this, some mutations are good because they produce new characters, and new characters lead to evolution. And then ability to express as Mendelian characters. Remember, in genetics chapter we talked about two, Mendelian characters and non-Mendelian characters. So all the Mendelian characters that we talked about, various characters expressed by organism, they are all because of the DNA. And the deviations of Mendelian characters is because of mutations, as we studied in our previous chapter, right? And what is the basic difference between DNA and RNA? As you all saw just now, DNA is double stranded. We said double helix. RNA is spring like, only one thread, single strand. And in DNA, presence of thymine, T is there, nitrogenous base. In RNA, presence of uracil, U. So you must remember this, union territory, U, uracil, T, thymine. Because sometimes in the first term exam, since it is MCQ, you may get question that in RNA, uracil will be there. Equivalent of that in DNA is, it will give you adenine, guanine, cytosine, thymine. So you must remember union territory, UT, uracil, thymine. Okay? And then absence of second carbon hydroxyl in the sugar. That's why we said deoxy, DNA, deoxyribose. At second position, that O will be missing in the hydroxyl group. Only H will be there. Whereas in RNA, it is ribulose. Means oxygen is not deleted, oxygen is present. So presence of OH group, hydroxyl group at second carbon. These are the three basic differences between DNA and RNA. And another one also Kadkol knows, that DNA and RNA is present in most of the organisms, including Kadkol. Whereas in viruses, RNA will be there. Some of the viruses will have only RNA. Of course, there will be some viruses having both DNA and RNA also. But exclusively, some viruses will have only RNA. Right? Next.
Last year it was told that only one of the DNA or RNA will be present in virus.
Now, evolution, new things, new viruses, new research. You must understand, Sandeep, science is not static. It keeps on. What you have studied last year will change this year. Okay? Unless until it is not proving itself. Right.
Sir, plasmids are DNA or RNA?
Pardon?
Plasmids, sir,
Plasmid, okay.
They are DNA or
DNA. We defined, no, plasmid is a circular DNA. In the definition only we said. Plasmid is a circular DNA. We defined, no? In the definition only it has come. Okay? Right. So thus, RNA is unstable. So RNA viruses, see, viruses in which RNA is present. RNA viruses like bacteriophages, tobacco mosaic virus, TMV. Okay? In this only RNA is present, and mutation will be faster and evolution will be faster, Sandeep. That's why I said viruses will be evolving faster compared to us. And DNA strands are complementary. I said, what is that complementary word? On heating, they separate. Look at that diagram. I'll expand that in a minute. In appropriate conditions, okay, they come together. In Griffith's experiment, some properties of DNA of the heat killed S bacteria, smooth bacteria, did not destroy. It indicates the stability of DNA. Remember, recollect from your memory, Griffith's experiment? We said heat killed S strain. Even though we heated the S strain, the DNA did not die. Why? It is stable. Whereas RNA is very much unstable. That's why it undergoes mutations easily. And whenever mutations are taking place fast, evolution also fast. So we said a tobacco mosaic virus, you can see the diagram of that. One is the bacteriophage, another is the tobacco mosaic virus. And the DNA replication you can see here. So look at that, double helix model, supply heat, and the DNA threads are breaking. Where they are breaking? In between the nitrogenous bases. Why so? Weak hydrogen bond is there. Yeah, weak hydrogen bond is there. So you get virtually two vertical strands like this, separated strands. And if we can synthesize this duplicate, so instead of two strands, we have four strands, two DNAs. So that daughter DNA goes into the daughter cell. This is how daughter cells also will be getting the same characters of the parental cell.
Both
So
the DNA will be same, no sir?
both DNA will be same, but complementary. Complementary means, like I gave the example. Basically, Javagal Srinath, Venkatesh Prasad, both are bowling fast. They are fast bowlers. But Javagal Srinath bowls in swingers. Ball pitches on outside off stump and goes to the leg stump. That's called in swinger. And Prasad bowls, not our Prasad, no? Venkatesh Prasad. Bowls out swingers. He pitches the ball outside the or off stump or off and middle and goes outside the off stump. So we say both are complementing each other. You must observe Sunil Gavaskar, Ravi Shastri, Sanjay Mandirekar commenting that both the Indian pace bowlers are complementing each other. Basically they are fast bowlers, pace bowlers, but one is bowling in swinger, another is out swinger. Here it is nitrogen base adenine, there it is thymine. Here it is guanine, there it is cytosine. But properties remain same.
There won't be any change, sir.
Properties are same, no? So that is the thing. And when we cool this, again, look at that, double helix reformed. So this is how the new cells will be getting DNA. So thanks to DNA replication, otherwise it would have been very difficult for us, right? And because of this DNA replication, Sandeep is able to grow. How? This is the thing. DNA is becoming RNA. That is, double helix thread is becoming single thread. So virtually, DNA is becoming RNA. We call this process as transcription. Don't get confused because both are similarly sounding. One is transcription, another is translation. Translation is RNA producing protein. Okay? And I am sure Sandeep must have observed. Under transcription, we are putting two arrow marks. Means what? Yes, reversible. In chemistry, you studied many reactions, no? Reversible reactions. So DNA can become RNA, RNA can become back to DNA. How to do it? Just now above I said, heat the DNA molecule, cool the RNA molecule, both the strands will join together. So for protein synthesis, we have to depend on DNA. There is no other way. Okay? And for the storage of genetic information. Now, when DNA breaks having genetic information, ultimately, Kadkol, where is it going? Look at that. Ultimately, our secrets are going to proteins. So only some time back, maybe 10 years back, let us say, if someone asks our Kadkol, what is your secret? Kadkol used to tell, my nucleus. But after some years, someone asks Kadkol, what is your secret? Kadkol used to tell, my chromosomes. Recently, may say 4-5 years back, someone asked, what is your secret, Kadkol? He used to tell, my DNA. DNA is my secret. But you ask Sandeep now, what is your secret, Sandeep? Sandeep says proteins. Why? Ultimately, all our secrets are coming to proteins. And without proteins, no life. Hence, our secrets are our proteins. Why we behave in a particular situation, in a particular way, because we have that type of proteins in our body. And who is preparing it? Translation. And how translation is formed? Transcription. And from where transcription started? It's DNA. Correct? So, what is important for protein synthesis? RNA. Without RNA, there cannot be protein synthesis. So, basically, is there any one RNA or many? As on today, it is observed that 3 types of RNAs are present in our body. One is the messenger RNA, mRNA we call it. It is just like a railway platform, right? mRNA, one single thread. Then the second one is rRNA, ribosomal RNA. Means, you know that ribosomes are responsible for protein synthesis. So inside that, the thread will be there, RNA, and that is called rRNA, ribosomal RNA. And third one is tRNA, transfer RNA. Now, if you look at the picture carefully, does it look like lavanga? In Kannada you call now clove.
Yes.
Yeah, just carefully observe it. Does it look like that? So we say clove shaped, lavanga shaped. So basically, mRNA, rRNA, tRNA. 3 types of RNAs are there in our body. So RNA was the first genetic material. Before we could find out, thanks to Professor Crick and Watson, we were all thinking RNA is the genetic material. It acts as genetic material as well as catalyst. Without that, no proteins. And they are essential for life process. They are responsible for metabolism. They are responsible for translation. They are responsible for splicing. Splicing means splitting, okay, dividing. And it is evolved from DNA. For stability, of course, DNA evolved from RNA, because RNA single thread, just now we said it is highly unstable because it is not saturated, whereas DNA is saturated. That is the thing. So, lot many discussions were there. What is the central dogma of molecular biology? Means how we are able to be alive. That was the question that was going on in many heads. What makes us to be alive? So somebody like our Prasad said, Lord Venkateswara. Somebody like our Gollal Gowda said, Veerabhadra Swami. But you know, somebody like Kadakol said, okay, that's okay. But basically, how we are able to be alive? And the answer is transcription, translation. These are the 2 processes that make proteins to be synthesized in our body. And once proteins are there, we express our traits, characters. Like I gave the example, if Prasad is very tall, he is tall because he has got proteins for tallness. And who is preparing the proteins? His RNA, his DNA, his nucleus, his cell. So we call this as central dogma. So the central dogma is proposed by our favorite, Francis Crick. And unfortunately, in 2004 we lost him. Otherwise, He was a genius. He was the authority for the DNA. Okay, and thanks to him that he could answer this central dogma. Dogma means puzzle, like question mark. With what we are alive? How is that living organisms are living organisms? How is that living organisms continue to be alive? This is the question. Okay.
So,
Oh yes.
so this RT-PCR test is this one also.
Yes, yes, exactly. I am coming to that. I am coming to next slide. There I will tell you. Okay. So, thanks to this gentleman, Professor Crick. And then, in viruses, as our Sandeep says, there is something called reverse transcription. In some viruses, flow of information is in reverse direction. Because some viruses will have, I said, only RNA viruses. So what they do, they duplicate the RNA thread and become DNA. That is what when Sandeep says RT-PCR, reverse transcription, okay, PCR, polymerase chain reaction. This PCR I will explain in 2 days. Next 2 days you will understand what is PCR, right? So since the RNA is becoming DNA, ulta is going on, reverse is going on, we call this as reverse transcription. And because of that reverse transcription, now proteins are being synthesized. So there are 2 ways. One is the straight DNA to RNA, RNA to protein. Another one, RNA to DNA, DNA to RNA, RNA to protein. Okay? So there are 2 ways that protein synthesis can take place. So overall, thanks to DNA replication. Without this DNA replication, we wouldn't have been alive. And there is one experiment called Messelson and Stiles experiment in DNA replication that I will take up tomorrow. Okay? We'll meet again tomorrow. Take care. Bye.